and say welcome. So welcome to our reproducibility colloquium. So my name is Eric, and I work with the Communications and Cyber Technologies Data Science Group. And I just want to welcome everybody to this colloquium. So first of all, I just want to give a little bit of background on who our group is, meaning who I work for, and then I'll talk a little bit about our learners too, and they'll introduce themselves. So yeah, we are technically an experiment station, a virtual experiment station, or part of one. Christina Reamer here is my boss, and we've got a few other members, including me and Renata, who co-taught this workshop series. And what our group offers, workshops. So we have monthly workshops on various topics. You can find our past workshops on YouTube. We also have a data science incubator program that is free or pre-funded for anyone who is in the ALVSCI acronym, which is primarily the College of Agriculture, Life and Environmental Sciences, but also experiment stations and extension. And this allows you to collaborate with us for short-term or preliminary projects. You get like up to 80 hours of our time where we work on a project together that eventually we kind of hand off to you, either because it's finished or because you're ready to like take over. So there's a lot of training involved. It's a really great process. It's very kind of informal to get started. So if you're interested, contact us and we can put together a project plan together. And all of the learners at the workshop are also definitely invited to do this. Think if you have an incubator project in mind that you'd like to work on us with. We also offer help with grant writing. So for example, if you're working on a grant and you need a data management plan, or you know that you have some data analysis that's going to happen in this grant and you want to get us involved early on, that's great. And we also can work with you on longer term funded projects. You can pay for a portion of my time or somebody else's time in our group, and we can work on projects with you. And we've got a bunch of previous projects that you can check out on our website for examples. And we also have drop-in hours that are virtual every Tuesday at 9 a.m. And all of that information is here on our website, which is datascience.cct.arizona.edu. You can see information about our drop-in hours, our upcoming workshops, the incubator program, including examples of past incubator projects we've done to get an idea if maybe that's a good fit for you. And then other kind of longer term projects that we've worked on. All that stuff is available on our website, and we encourage you to contact us and let us know if you're interested in working with us. So what we did here was what we're doing today is we're sort of kind of like celebrating the culmination of this reproducibility and data science in our workshop series that we offer every fall by application only. So the folks that are presenting today are had to apply and were accepted to this and went through a variety of topics. We went through lessons that went from organizing a research compendium, talking about reproducible reports and manuscripts, version control with Git and GitHub, data wrangling and programming with R, and a bunch more stuff as well. Archiving data and code as well. And today, our presenters are going to show us, some aspect that they learned from this series of lessons and how they kind of applied it to their work. So welcome everybody. And I'm going to we'll have this contact information here, and I'll share this as well in whatever venue this video ends up getting shared on. We'll probably put it up on our YouTube channel, and it'll be in the description. So I would like to, we'll start with Nastasia, and I'm going to stop sharing here, and I'm going to make sure to allow people to share. There we go. So if you want to take it away from your hotel lobby, thanks for coming.
Yes. I will send. Okay. So do you see my
Yep, looks
slides
great
now? Okay. Well, I'm Nastasia, so I work on the Valley Fever. I'm based at the Environmental Science Department. And the valley fever is endemic to Arizona, California, and some parts of Mexico and Brazil. But yeah, here, because I'm with the University of Arizona, it's funded by the Arizona Board of Regents because it's a problem of Arizona. And so I'm not working on this alone. There is like a PhD student, so Brooke Baya, me, I am the postdoc. We have a professor associate, Rob Bruce and Craig Rasmussen, and my supervisor, John Shorover. And so valley fever is caused by a fungi, the Coccidoides immitis or Coccidoides posodosai, and they are found in the soil. So the general aim of the project is to find correlation between the presence of Coccidoides in the soil and soil and climate properties. And for the colloquium, I just wanted to focus on a small study we have, which is on seasonal variation of the presence of Coccidoides. And so so the data set for this small study, it just result of quantitative PCR. So PCR is like an acronym for polymerase chain reaction. And this is basically DNA replication. So we go monthly in a site in Tucson, and we sample soil, and then we extract the DNA, and then we have to amplify the DNA so there is enough DNA so we can analyze it. And we check a specific gene, which is specific to Coccidoides. And yeah, so in brief, that's the method. And we started this since November 2023. So we'll almost have a year in a month, in a couple of months. And we sample 6 rodent burrows because there is like a theory that said that it's probably linked to rodent activity. So that's why we have this focus on rodent burrows, but we also have like 5 transects, which are like a soil, like some area which are far from rodent activity, so far from rodent burrows, to see if we would be able to find it as well, like far from rodents. And the final like data set is like the template in between the big template and the figure. And it's really small. I just have like each of my burrows and each of like my location of soil, and I have a data which is either 0, 1, or 2, and then I have the months. And this is because for each soil sample, we do 2 DNA extraction. So I can either have one where it's present, it can be present in both DNA or in none of the DNA. And that is because it's very like heterogeneously distributed in soil. So even like one same soil sample, like you can do multiple DNA extraction on it and don't have the same results. So that's why. And yeah, so when I acquire the first data, I put it in an Excel spreadsheet, and I do like the first data processing in Excel, because I need to have also like a lot of control for DNA extraction, control for the qPCR reaction, and I can only use the data if the control has passed. And this is done in Excel, and then when I'm happy, I can trust the data. I just have the summary in a CSV file, and then I do the figure on R. So yeah, what I wanted to focus on for the makeover and my take on the workshop was to use version control and be able to work on my code remotely as well, like on a different laptop, and be able to share my code with the other members of my team and my lab, because we have other projects and can be beneficial as well for them to know how to do analysis and make figures on R. And I wanted also to improve the organization on my files and the script reproducibility, and also the efficiency for writing my script in general. So I can show you in brief how it looked like before. So you can see on top like where was the project, and I have like so many like subfolder, and my organization was really inconsistent. Like I just figured this out that because I have like a folder for data analysis, but for that study I just had folder was not in the data analysis folder, which is the folder that was dedicated for like our project. So there was that. And also, so I wouldn't save my old version of the script. I would just like keep one script and save that one. And which, yeah, I didn't know otherwise. The only remote access I have is iCloud. I use iCloud more for like a backup, but you can also access your file. But then it's really not convenient for like writing code. Like if the machine doesn't have OR, it wouldn't be able to open in a OR script. Maybe I could use like another text editor, but then it's just like very hard. Like you don't have the color differentiation for the function and so on. So, and when I share codes, I would share by email. I wouldn't have documentation. And also you can see like at the top of my R script, like I would always have this bulky list of packages. And I wanted to learn how to have just like one line of code, which is something then I've learned through the workshop. So I can show that. The after, so now I made like a folder for my R project into like my work folder. So it's a bit more like straightforward, and then I can just name them according to the research project I am. So I know to which project it relates to. And I also managed to do like a script in which I have all the packages that I usually use. And they are like small packages. I wouldn't put like a big package because otherwise like a big package that I just like use for specific things. Like I'm thinking microecho, which is like huge. So this one I wouldn't put it in there because it's not worth to have like a package that takes like 5 minutes to load if I don't use it. And this is only for like microbiome analysis. So this one will be like separate, but for the other one, I just made like a script, and then I have like a line on top of my script for the project where I just source that script. And I think that's really cool. Like for me, it's like very tidy now. It's just one line. I also have like a line to like show the one that are supposed to load. And I can just compare with the one which loaded. So I'm sure that everything worked, especially if I add like a new package. And yeah, so that script with the packages is at one place. If I find like a package for like data analysis, which I like and happen to want to use it, I can just add it in it. And yeah, that's it. So that's really cool. Then I also standardize the setup of my R environment. So with this line, and then so for uploading the packages and then to read the data. And also, so because I used our base before, so I have like a few lines at the top, which is only for like setting up the working directory, which is needed when you use our base. And yeah, I just wrote down that if I use our studio, I don't need to run these lines. And so I think it's cool, even like someone may use our base, so they will have this line if I share the script with them. And now I will have like a README file as well, which you can see on the RStudio, like the pan of the files. And I can use Git for version control. So same, I think, yeah, you see on the file panels, and I can use GitHub, which I think I opened the Git. Yeah, so you can see I can use GitHub with it. So that's really, really cool. So, oh, sorry. So yeah, the conclusion for me, that was, I was super happy that Git and GitHub has been demystified. I always wanted to learn how to use them, and I just thought it was way more complicated. But no, it's okay. I just need, I guess, to like still practice, like to remember the commands for Git. And I also do a lot of statistical like significance tests and other like analyses which are very repetitive. And for that, I will definitely implement the iteration like commands that we've learned. So yeah, that will be my next step for me. So yeah, thank you for listening to me. And if you have any question, you can let me know.
Awesome. Thank you, Nastasia. Yeah. Are there any questions? I have one. I'll ask as you've put your work on GitHub, and you mentioned in the beginning that you're hoping to share your code with your lab, your collaborators. How has that gone? Have you gotten folks to interact with your code on GitHub in any way, to look at it, to comment on it by leaving an issue, or is that still a work in progress too?
It's definitely still a work in progress. So we are like more like chemists, geochemists. So I think there is like one PhD in my group that works with Python. I think she's been doing a bit of modeling, but yeah, other like for the other member, it's maybe a bit like unknown territory, but it would be really cool to be at that stage where we have maybe like a repository for our lab and we can put the script like, significance testing is like very basic. So just have that and they know, oh, okay, for that test, that package is like good for that and things like that.
Yeah. Cool. Well, if you ever need help convincing somebody, or help setting that up for your lab, let us know for sure.
Oh
Any
yeah.
other any other questions for Nastasia? All right, and thank you again. And we'll go to Margaret. Are you there? Would you like to share your screen? Present.
Yes, hi. Yeah, I'll go ahead and do that.
Sure.
Let's do entire screen. Where am I? Cool. So can you guys hear me okay?
Yep, sounds good. Looks good.
Lovely. So my project, this is chapter 2 of my thesis that I'll be presenting on today. It's an analysis of behavior of Tucson bobcats around roads. And so looking at just, do they avoid roads? Do they cross roads more or less frequently than would be expected if they were just moving around randomly? And then are they using crossing structures like culverts or bridges to cross roads, or are they just crossing roads wherever they come across them? And then does their speed vary as they approach roads? Are they slowing down? Are they speeding up? Those kinds of behavioral questions as well. And so, oops, there we go. So the data I'm using is a roadmap along with bridges marked. So those are marked in blue against the red of just the roads around our study area. So this is like the west half of Tucson roughly. And then I'm using GPS collar data on bobcats. So those are kind of the dots in the middle. That's all the bobcats, but we're splitting it up by individuals, so we get individual home ranges. And then we're using a continuous time movement model in R to figure out where the bobcats likely were between the dots. And so to sort of create like a path, which you can see in blue on the right. Blue is the path, red is the roads, and then the black dots on the part to the right is where that individual bobcat crossed roads. So you can see there's quite a bit of road crossings, especially in the middle of the range. And so that's sort of the, quick overview of my project. And then, so sort of the issue is I really just had one big old script to run through the whole analysis, and I hadn't gotten it to run through each bobcat individually. Like I would have had to go into the script and change which Bobcat it was pulling one by one, and there were like 36 Bobcats, so that would have taken a long time. And so I wanted to run it through the HPC and tell it to pull each of the information from each of the Bobcats individually, so I could just send it to the HPC, have it run, and not have to go in and change things once each analysis was done. And so also each part of the script had like different outputs. And so I was wanting to split it into several different scripts. In addition, some Bobcats are range resident and some individuals were not because they were dispersing juveniles. And so the analysis was going to be a little bit different for them. So I needed to do the whole analysis. I had needed 2 different scripts for the whole analysis, one for range resident, one for non-range resident. So I needed to do some splitting up and getting stuff ready to run on the HPC. And so one of the things I did was I organized my analysis scripts better. I really liked what we talked about, having like scripts numbered 1 through whatever. That was really useful for me in organizing my stuff. And so like I have 1A for range resident and then 1B for non-range resident. So that's sort of how I sorted it out. And I feel like that was pretty effective. And I put, so when you run on the HPC, you need like separate bash scripts for each R script that you're going to run. And so I put the bash scripts and the outputs in their own folder. So I wasn't like mixing things up. And then within the bash script, I'd reference the R script, which was in a different folder. And then I organized my results into folders a little bit better within the larger results folder. So I feel like my file structure in general is improved. And then, yeah, just reflections. I feel like numbering is a good idea and one that hadn't really occurred to me. And I feel like I'm going to apply that to my other chapter as well, which has a lot of data wrangling and a lot of working with different data sets. And so it would really help if I could, with reproducibility, if I could say, here, you got to do this one, and then this one, and then this one, and then this one. And so I'll hopefully get around to doing that at some point. That would be very helpful. And then, yeah, just as you can see, like this is my other chapter. I have a lot of different folders and a lot of different scripts within folders and different analyses that I'm doing. And so streamlining all that is probably a next step for me. And yeah, that's it.
Awesome. Thank you for sharing. It's a very interesting project. Questions, Margaret? I have a question. How did you learn to use the HPC?
So my advisor taught me because he knew I was going to have to be using it for this project. And so he sort of gave me a quick crash course and I wrote everything down that he said, but then I had to keep going back and being like, hey, help with this, help with this. What did you say about this? So it was definitely a whole process, but I'm really glad I did. And now
Awesome.
that I have like the bash script outline, like I can just change it and I can just run different things now that I know how to do it. It's very convenient and I can help other people in my lab with it too. So it's definitely been
Awesome.
very helpful. Yeah.
Very cool. That is a superpower indeed. Awesome. Other questions for Margaret? Okay. Well, we'll move on to our last presenter, Danny.
Hey, yeah, so I'll just reintroduce myself. I'm Danny, I'm a first year master's student, so I will be presenting an independent research project or part of an independent research project that I'm working on for a class. And
Cool.
yeah, let me share my screen. Entire screen. Okay, so I'm just gonna, so I have things kind of in some different places. I was gonna show you my R and then the GitHub repository that I made, and then the PowerPoint is more for the presentation that I'll be giving for my independent research project. So basically the project is going to be looking at the RFIA package and kind of presenting the functionality of it. Because I will be working with FIA, which is Forest Inventory Analysis data for my master's thesis project. And if you're unfamiliar with the FIA program, it was mandated in 1928 by Congress as like a periodic inventorying of different forest units. And then 1998, with that farm bill, they turned it into an annual inventory. And so all of that is now, all that data lives in the FIA data mart. And actually I can go to this is the actual website for it. And if you were to try and download this whole database, I think it would probably crash your computer and take forever. So that's probably not wise. So instead, the creators of this package, and I think everybody who works with FIA data, recommends that you only download the data that you need and subset it out. But before I did any of that, I first set up version control with Git. And I used the use Git function because when I made the project, I did not check that little box. I was like, use Git. So I used the use this package to do that. And then I linked it to my GitHub for the project, which is here, and made it so that I could be pushing and pulling from there. And I don't have too many things in here right now, but I did just commit a change to it recently, which was fixing the formatting of my README. So anyway, yeah, so far it's been a lot of data wrangling and looking at the functionality. And so currently the package, I think it was developed as and published as someone's master's thesis project back in 2020. And since then, it has been taken, the package is off of it's no longer a CRAN package because it's fixing some issues. And so I couldn't just install packages RFIA. I had to install the development version using DevTools, install GitHub, and then that's how I got the package. And then there were a lot of useful things when I was going in and downloading a subset of the data. It had this nice little note of setting the timeout option to an hour because R automatically stops downloading after 60 seconds. So instead, I set it to download for an hour, and I was able to get all of the data from the state of Arizona. So that's what I did. And then from there, I explored one of the basic functions of it, which is just looking at like trees per acre estimates and tree abundance estimates. And I can show you, and I could look at that by species. I could look at it by size class. And this is some of the code that I used for those. And then I was able to make some plots, and I'll pull those up. So it's kind of a goofy looking one at the moment here, but this is trees per acre by species for the first 5 in that data frame because there are a ton. So I just pulled the first 5, which is Arizona white oak, common or 2 needle pinyon, gamble oak, ponderosa pine, and Utah juniper. And this is just trees per acre and by size class down here. So you can see there are a lot in these smaller size classes and then none once we get up into these big ones. And then I wanted to look at it by county. So then I went here and grabbed the county shape files and then loaded those into R using this code at the bottom here in the SF package. And these are the maps that I created them for every year in between 2005 and 2021. Not before that, because that's when all the periodic data was, and when they were doing the periodic data, they were using like different sampling methods. And so after that 1998 is when things got more standardized and we can look at them year to year. But one of the issues with FIA data and the FIA program is that the Forest Service is pretty limited by funding. And so they can't get to all of their plots in every like forest unit every year. And so in the West, I think we get revisits every 10 years. And on the East Coast, it's more like every 5 years. And so that's definitely one of the limitations of using this package and the FIA program is just you have those big gaps in between resampling. But these are tree abundance maps by county, and you can see in 2025, I probably could change the coloring of this because I think yellow makes me think that there are fewer, but in this scenario, there are more. And so maybe I would want to switch the type of color that I'm using or the scale. But yeah, you can see up here in northeastern Arizona, a lot more trees compared to in 2021. We kind of went down by, you know, it looks like 100 or so. But yeah, so that is basically what I did. I definitely am also grateful to have Git and GitHub demystified because I'm the type of person who will often not close a tab forever because I'm worried that it's going to go away forever if I close the tab. And now having, you know, the security of those things being up on GitHub and having the different versions of them is a lot more helpful. And I also did organize my files a little bit better. I like put all my data in the data file and I put my figures in this file as well. And yeah. So.
Awesome. Thank you. Very interesting project. And that's a great example of all the GitHub skills that you guys have gotten. Even if you don't ever put any of your own code up on there, you have now the ability to find R packages on there that maybe no longer exist in other places. So that's a great example of that.
Yeah.
Cool, thank you.
Yeah. And I will mention one more thing is I will be working with GitHub a lot on the forest time R package that's being created, and Renata is the main author of that code. And so, yeah, it's very helpful to have the background knowledge of the R FIA package and what's already out there, and then looking at forest time, which is being developed, and that's going to be like annualizing things. And yeah, it's all good.
Awesome. Yeah, and if anybody else is interested in making an R package, we can help you with that for sure. You can learn how to do that. So, any questions for Danny? Okay. Well, thank you again. Is there anybody else that wants to present something? It's okay if you don't. I didn't know if you're just here to watch and support your fellow learners or not. So, well, okay. Thank you all for coming. And I will again just remind you all, well, first I'll say it's been wonderful. What an awesome group. It's been really cool to have such a group that's been really involved and vocal and like is not afraid to unmute and ask questions and get through difficult, weird GitHub bugs and things like that. So it's been a good learning experience for me and hopefully all for you. And we will be offering this workshop series next fall. And so please like recommend it to your friends, lab mates, and so on. And yeah, get in touch with us if you need anything data science related, have questions or all that. I'm going to be sending out a follow-up email to everybody who was accepted into the fall workshop series, which is a lot more than that's in this room, with some follow-up things, links to resources, reminders about like you'll still have our Slack channel, our private Slack channel, you'll still be able to use that. And most importantly, a survey. So please fill that out just to like give us some feedback on how things went for you and what kinds of topics you were most interested in and most likely to apply to your work, because we're going to be making changes in the future and just want to make sure those changes to the course like align with what everybody thinks about it. So yeah, with that, unless there's any final comments or questions or anything, we'll wrap things up. And thank you again so much for being here, everyone, and great job. Awesome work.
Thank you, Eric.
Yeah,
All
thanks,
right.
Eric. This has been super helpful, and I look forward to collaborating more in office hours, maybe.
Yeah, absolutely. Yeah, we'll see y'all at drop-in hours. I'll see you on Slack. We'll see you at workshops in the future. Keep an eye out on our future workshops. Everybody that presented something, I thought, ooh, this workshop would be good. Oh, they might like this workshop. Oh, they might like this workshop. So keep an eye on our future workshops. Check out our past ones on our YouTube channel. Yeah, let us know if there's anything missing there that you would like to learn more about. We can kind of offer workshops on request as long as it's something that's like vaguely in my wheelhouse. I can come up with some materials and learn some stuff so that I can teach y'all. So, all right, well, have a great day, and yeah, thanks again, and we'll see you around.
Thank you.
Yeah,
You.
bye.